Okay. So what we'll do is we'll start and just go through some anatomical features of the male reproductive system, and then we will move on and talk about the physiology of male reproduction, which is a process called spermatogenesis, which just means to make sperm, right? So, as we discussed on Tuesday, the gonads for the genetic males are what we call the testicles or the testes. The testes are kind of egg-like structures that sit in an epithelial sac that's called the scrotum, and it's within the testes that sperm are both made. And then I'll say this as again when we get to the slide that addresses it, but just maybe make note of it in your head right now. So sperm are produced in the testes, and then what they do is they migrate through this duct system. So they're produced in the testes, but they mature in what is called the epididymis. Okay, and you can see the epididymis is this structure here that is attached to and a part of the testes, but is histologically distinct from the testes. Okay, so again, sperm are made in the testes, but they mature and become capable, meaning that they would be capable of fertilization. They can swim within the epididymis. And then what happens is once sperm are capable or mature, they leave the epididymis, and they leave the epididymis through a series of ducts, right? And the epididymis is technically speaking the first part of that duct. So the duct system would include the epididymis, this structure right here called the vas deferens, or excuse me, the ductus deferens, which consequently is the tube that if a genetic male decides that he would know he no longer wants to be fertile or produce viable sperm, a very popular and easy procedure is what's called a vasectomy. And during a vasectomy, the vas deferens or the ductus deferens, which is again just another name for the vas deferens, is somehow severed, right? So there's a cut and tie technique, there's cauterization, there's clamping, several different ways you can go about doing this. But this is the part of the male reproductive system that is disrupted during a vasectomy. From the vas deferens, we then have what's called the ejaculatory duct, which is right here. And you can see a sperm is traveling through this duct system. We're going to talk about these accessory organs, but this duct system passes by several different accessory structures, right? And so, in those accessory structures, such as the prostate gland here, the bulbourethral glands, they produce substances that are very important to nourish sperm and to essentially allow them to be viable if and when the sperm get into the female reproductive tract. And then after the ejaculatory duct, we have what's called the urethra. So the urethra for males is relatively long. The urethra is something that differentiates the urethra from males to females, is that it acts as a passageway for both sperm and for urine. And that has some important repercussions when it comes to sperm viability, right? Sperm, like all cells in the body, don't particularly like a pH that deviates too much from physiological pH, and they really don't like an acidic environment. And urine, by and large, tends to be acidic, right? So it's not, urine by its very nature is not particularly good for the health of sperm. And so, as a species, we have adapted to have certain substances, which we'll discuss, that help to kind of flush out the urethra when ejaculation takes place, and to help kind of neutralize the urine so that it doesn't damage the sperm. Okay, and so again, those accessory glands that we're going to be going through and looking at in pretty good detail include what are called the seminal vesicles, the prostate gland, and then the bulbourethral glands. And what these structures do is they make secretions, and those secretions are there, as I mentioned, to nourish the sperm, to help neutralize pH, both from urine, and then also the vagina, the female reproductive tract, is a relatively acidic environment. And so the pH or these fluids help to neutralize and protect the sperm from that environment as well. Okay, so what you need to know about the scrotum. The scrotum again it's the epithelial sac that holds the testicles. The scrotum play an important role in helping to protect the testes and also play an important role in keeping the testicles at a slightly lower than normal body temperature. Um, so sperm production takes place best, if you will, at slightly lower than normal body temp. And so the scrotum, along with the associated muscles, which are specifically the dartos and the cremaster muscles, essentially hold the scrotum in place. So if it's cold outside, those muscles can move the scrotum closer to the thighs in order to increase the temperature within the testicles. If it's warm, the opposite happens, right? The scrotum can move the testicles away from core body temperature to help cool them off. Okay, so now we're going to get in and look at the micro anatomical properties of the testicles and the duct system. So this is showing you a section through the testicle and part of the epididymis, right? And what you'll notice, and hopefully you've seen this when you have looked at the slides associated with the lab and looked at the testicle is made up of a series of tubules, right? So they're these circular tubules, and those tubules are what are called the seminiferous tubules. The seminiferous tubules are made up of two very important cell populations. The first that I have listed here are what are called the suscentacular cells. So the suscentacular cells think of them as the supporting cells. They are there to help support the spermatogonium, which are the germ cells that are going to undergo differentiation and become sperm ultimately. They're there to help that process take place. And they do so in several different ways. So, one important characteristic, and I should mention just kind of something that might help you remember suscentacular cells is that suscentacular, they're also called nurse cells. And I believe that root of the word suscentacular uh, means to nurse or to take care of. So suscentacular cells have receptors for follicle stimulating hormone. And in the presence of follicle stimulating hormone, suscentacular cells secrete something called ABP, which is androgen binding protein. Okay. What ABP does is it binds testosterone. We talked on Tuesday about kind of briefly about hormones, and I made mention of the fact that hormones play an absolutely critical role in both male and female reproduction. So for male reproduction, the primary hormone that's responsible for viable sperm production is testosterone. When testosterone levels get too low, and for that matter, when they get too high, but when testosterone levels are out of range, sperm production is negatively impacted pretty dramatically. So suscentacular cells, when the anterior pituitary gland secretes FSH, that will get into the bloodstream and it will bind ultimately to receptors on the suscentacular cells. And those cells, again, will secrete ABP, which will hold any testosterone it'll hold that testosterone in place. Okay, kind of keep that in the back of your mind when we get to spermatogenesis. Another important cell type that we find that within the seminiferous tubules are what we call interstitial cells. These cells are also called Leydig cells, Leydig. I will use the word interstitial, but just for those people who may be taken or read this information in other textbooks. So what interstitial cells do is so they have receptors for luteinizing hormone. And when luteinizing hormone is around, the interstitial cells will produce testosterone, right? So the testosterone is made by the interstitial cells in response to luteinizing hormone, and then is held in the vicinity by ABP from the succentacular cells. Okay. Um, just to also to make mention, the vast majority of the testosterone that is responsible for male reproduction comes from interstitial cells, but it is worth noting that they're with the adrenal glands also make androgens, right? They don't make androgens, they don't produce levels that would be high enough to support reproduction, um, the production of viable sperm in this case, but they do secrete, they are responsible for making uh, and secreting some testosterone. Okay, let me see if there's anything else on this slide I want to make mention of. I don't think so. Okay. So this is just another cartoon image of a cross section through the testicles and the epididymis. Um, as I mentioned, the epididymis is where sperm will mature, where they kind of learn to swim. Um, Between as the sperm migrate from the seminiferous tubules to the epididymis, they pass through what's called the rete testis, which is just this, it's kind of the bridge between the two areas. And another thing, and I mentioned this on Tuesday, but when we talk about the way hormones, the endocrinology of male and female reproduction, but we're talking about male reproduction right now, um, the vast control mechanism is negative feedback, right? So actually, let me back up 1 second to this slide. So because I didn't talk about inhibin. So in addition to ABP, succentacular cells make and secrete inhibin. And inhibin is a hormone that helps to inhibit um, ultimately that the FSH LH spikes, right? And inhibin is succentacular cells are stimulated to secrete inhibin when testosterone levels become really high, right? So ultimately, this hormone will shut down LH and FSH production and and lower testosterone levels and keep testosterone within that parameter that's required for or the parameter that best supports sperm production. I should say healthy sperm production. Okay, so I like this little graphical depiction because it shows you the negative feedback, right? And it also kind of just outlines in a really simple way the steps by which sperm is produced when it comes to endocrinology. So GnRH for both males and females is what starts this process off. What ultimately um, that's the hormone that's ultimately responsible for the production in males of viable sperm, and in females, that's the hormone that sets off the the cycle by which the menstrual and the ovulatory cycle. So GnRH, if you, and I point that out because GnRH, it's kind of what we consider the puberty hormone, right? It's a hormone that you see that is relatively, it's always secreted, but at relatively low levels in childhood. And then based on a variety of different factors, but somewhere between the ages of, she's anymore now 9 and 17, GnRH levels increase. There's kind of this dramatic increase in GnRH. And what GnRH does, and it comes from the hypothalamus, is it talks to the anterior pituitary gland and it tells that gland, to that endocrine gland, to secrete FSH and LH, right? And again, just for the purposes of being redundant, because I think it's helpful with this class, FSH binds specifically to the succentacular cells within the seminiferous tubules, and luteinizing hormone binds to the interstitial cells within the seminiferous tubules. In response to FSH, the succentacular cells will secrete ABP, right, which is androgen binding protein. And androgen binding protein is able to hold testosterone, and luteinizing hormone will stimulate the interstitial cells to secrete testosterone, which is required for the process of going from a spermatogonium to a mature sperm. So testosterone is the hormone that stimulates the spermatogonium, the germ cells, the stem cells, um, the sperm stem cells to undergo differentiation. All right. And then, hold on, let me put my coffee down. Then, as we just discussed, when levels of testosterone become really high, that information is detected by the subsentacular cells, which secrete inhibin. And then inhibin will ultimately shut off the GnRH, LH, FSH spikes. And so it'll, at the end of the day, shut down FSH and LH production, or slow it down, I should say. And then testosterone itself also will negatively feedback and decrease LH levels and FSH levels. Okay, let me see if there's anything on this slide that I didn't already say. I guess the only other thing I would mention that's on this slide that we haven't really talked about yet is that I think I might have mentioned on a Tuesday, but testosterone is the basis, the hormone that is the basis for libido, which is sex drive, in males and in females. Testosterone, and I guess this is a good time to mention that although from a profile standpoint, testosterone is the main genetic male hormone, and estrogen and progesterone are the main genetic female hormones, both males and females have estrogen, progesterone, and testosterone. It just, depending on the presence or absence, I should say, of the Y chromosome, those endocrine profiles differ between the different sexes. Okay, and again, I just like these slides, these little graphical depiction of feedback loops, because I think they help you to visualize what the hormones do, where they come from, and then how, when levels become too high, those hormones can feed back and shut the process down. Okay, so now we're going to go walk through the process of spermatogenesis, right? So what you're seeing in this picture is really close up, a cross section through a seminiferous tubule. Okay, so shown in purple here, these are the subsentacular cells, or those nurse cells, right? Up here, which you can't really see very well, maybe this is not in your viewpoint. Up here are the interstitial cells, right? So the nurse cells, again, are there to help support and nourish the production of sperm. So the differentiation of going from a spermatogonium, which is a sperm stem cell, to a proper sperm. And they're also there to hold testosterone in close proximity to those cells that are differentiating into sperm. Okay, so here are the subsentacular cells, here are the interstitial cells, and then what's shown in teal are the cells, the stem cells up here that ultimately, where's my cursor? There, that ultimately are going to differentiate into sperm. So the stem cells are, again, called spermatogonium. Anytime you see onium, right? O-N-I-U-M, like oogonium, spermatogonium, that tells you that we're talking about a germ cell. And what a germ cell is, if you can't remember from biology, a germ cell is a slightly differentiated stem cell. So it's a stem cell that is already kind of how can I say this? It's already gone through enough differentiation that it can't become any cell in the body, but it's the precursor cell for whatever, in this case, a sperm, right? So features and characteristics of spermatogonium. So spermatogonium are diploid cells, right? They're just kind of your standard diploid cell. And by that, I mean, they have 46 chromosomes, and histologically speaking, there's nothing aside from being a stem cell that's really noticeable about them. But when testosterone is around, spermatogonium will start to undergo, meiosis, right? Oh, actually, let me back up a moment. So before we get into meiosis, The spermatogonium itself, because it's just a normal diploid cell, is of course undergoing mitosis, okay? And mitosis just being the process of replication. One of the cells that that results from that mitotic division is going to be is kind of going to be the cell that is destined to undergo differentiation and become a sperm. And then the other cell is just kind of a reservoir, right? And as far as which cell is going to go undergo differentiation and become the sperm, which one is just going to be an exact duplicate of the spermatogonium, just another spermatogonium, is we don't understand. There's really no way to predict it. So anyway, that's just to describe this little part of the picture right here. So as soon as we have the mitotic division in this determined cell, or this, I'm trying to find a more creative word for determined. But anyway, I'm just going to continue to use it until I can come up with something else. So this determined cell, is what we call a primary spermatocyte, right? A primary spermatocyte, histologically speaking, looks very, very similar to a spermatogonium. We just give it a different name because it is the cell that's going to go through meiosis to become sperm, okay? So the primary spermatocyte is a diploid cell, and this diploid cell starts the process of meiosis, okay? So it goes through meiosis, and we already talked about the steps of meiosis. And so we know at the end of meiosis 1, what we have are 2 cells that are haploid cells, right? Which this is telling you 23 chromosomes that we're talking about haploid cells, but they are haploid cells that are non-functional because the chromosomes are stuck to the sister chromatids are still stuck together, right? Remember, in meiosis 1, the centromere doesn't break. And so these are technically haploid cells, but they're haploid cells with twice the amount of genetic information that they need, right? We call these haploid cells secondary spermatocytes, okay? And then it's these secondary spermatocytes that then go into meiosis 2, which is just the process of, it's a reductive type of reductive division. And it's the meiosis 2 is literally from a process standpoint, the same thing as mitosis. And what happens is the centromere gets split and those sister chromatids are separated into individual chromosomes and divided out into 2 cells each. So at the end of the day, after meiosis 2 is completed, as we always see when meiosis 2 is completed, we will have 4 cells that are haploid cells, functional haploid cells. And because of all of the steps that we discussed when we went through meiosis, each one of these cells is going to be genetically unique from one another and genetically unique from the parent cell from which they arose, okay? So we call these functional haploid cells spermatids, right? You'll notice from this picture that spermatids do not really look like sperm, right? They still look a whole heck of a lot just like a cell, kind of your standard cell. So what happens from the spermatid level kind of and then on out in this process is that the spermatid, it's all a matter of kind of refining this cell and making it so that the cell is capable of being motile. Okay, so as the spermatid continues to migrate in toward, just to give you some perspective, this is the lumen of the seminiferous tubule. So as this spermatid is migrating or making its way toward the lumen of the seminiferous tubule, it's developing physically as far as it's developing what we call an acrosome cap, right? So the acrosome cap is a cap that contains digestive proteins. And what happens when the sperm bumps up, if any of these sperm are ever able to encounter an egg or an oocyte, that sperm will bump up against the oocyte in an attempt to break the acrosome cap. And then that acrosome cap, when it breaks, will release the digestive enzymes that then can chew through the wall of the oocyte to help the sperm penetrate that egg. The other thing that we see as spermatids are making their way toward the lumen of the seminiferous tubules is that they lose all of their this cytos. So this was cytosol out here, right? You can see the cytoplasmic remnants on the sperm here, and then you can see once sperm look like what we call sperm, those cytoplasmic, that excess cytoplasmic material is gone, right? Because this essentially, again, sperm, the main name of the game for sperm is motility, right? They're basically just little missiles that contain DNA. And so they, as far as, again, because structure determines function, because functionally they need to be able to move, anything that weighs them down or makes them kind of ungainly is going to inhibit their ability to move. So, and I bring all these kind of like maybe seemingly subtle, nuanced points up because when it comes to fertility, any little thing, when if we're talking about male infertility, one of the main things that a physician or I should say a provider will do is look at sperm under the microscope if somebody is struggling with fertility to see if the sperm look normal or not, right? And if you see sperm that have a lot of this excess cytoplasm still left, that tells you that the sperm that are produced are going to be not great swimmers, right? They're just going to be again kind of ungainly. All right. So by the time the sperm get to the center of the seminiferous tubules, and where they're going to go from the seminiferous tubules again is through the reticestes and then into the epididymis. What they're doing in between or as they make their way to the epididymis is they're just kind of learning how to swim, right? They're practicing, if you will, using the ATP to propel their flagella, right? So the tail of the sperm is called the flagella. Sperm are the only flagellated cells in the human species, right? So if you're genetic female, you don't have any flagellated cells. Yeah, I think that's all I want to say on this side. I'm looking to see if there's any other little detail that I guess I do expect well, you should know it for lab, and it's pretty basic, but I do expect you for lecture and for lab to know the basic anatomical parts of a sperm, right? So it's important that you know that the tail of a sperm is a flagellum, right? The mid piece here, which is kind of the neck of the sperm, contains a ton of mitochondria, right? Because again, sperm are basically DNA missiles, and moving this flagellum takes quite a bit of energy in the form of ATP. So that mitochondria is there to generate large amounts of ATP to be used for that process. And then the nucleus, of course, is going to contain the DNA, a haploid set, chromosomes, and then the acrosome cap we already discussed contains digestive enzymes. Okay, so here's just another slide that you can use to help you visualize the purpose by which sperm go from being spermatids, right, within still being held by a sustentacular cell to a functional sperm. So when it comes to the fluids made by those accessory glands, here's what I want you to know. So the seminal vesicles, well, actually, let me back up a moment. So the word semen is a blanket term used to describe a fluid that contains sperm, right? So what semen is made up of is what we call seminal fluid, right? And seminal fluid plus the actual cells, which are the sperm themselves. Okay. So what seminal fluid does, as I mentioned a little bit earlier, seminal fluid helps to nourish and protect the sperm, and it also gives them a medium to swim in. Okay. The seminal fluid itself is, of course, made up of secretions from the seminal vesicles, right? The bulk, in fact, of seminal fluid comes from the seminal vesicles. But in addition to the fluid from the seminal vesicles, we also get fluids from the prostate gland and what we call the bulbourethral glands. So seminal fluid is an alkaline fluid, right? As I mentioned, the bulk of seminal fluid comes from the seminal vesicles, 60 to 70%. And it's an alkaline fluid that the alkalinity is important. You'll notice as we go through the next two secretions that they're all alkaline, really. But having that higher pH helps to neutralize the acidity of the female reproductive tract. So as I mentioned, the vagina is a very acidic environment, relatively speaking, to the pH that sperm would like to be in. And so the seminal fluid helps to neutralize that environment and give the sperm a chance to swim up the vagina and through the cervix, and then ultimately into the fallopian tubes. Kind of more on that to come. Another important characteristic of the seminal fluid is that it contains fructose. And that's really important because fructose is required to nourish the sperm on this journey, right? Sperm itself, when we get to the development and heredity component of all of this material, we'll talk about the fact that sperm have to be able to, so when if and when fertilization takes place, it can happen within anytime within a 3 day, a 72, and even 3 to 4 days in some cases, but really a 3 day window, right? And that's because an egg, once ovulated in the female reproductive system, is only viable for 24 hours, right? So sperm has to be able to make its way from the epididymis through the duct system. Well, first of all, has to kind of be able to withstand hanging out in the epididymis for a bit, right? And then once ejaculated, making its way, out of the male body into the female reproductive tract, swimming up to the fallopian tubes, and then sitting there and waiting to see if an egg will float down, right? So it's a longer journey for the sperm have to be able to kind of have some staying power. And so having that fructose, that nourishment, helps to ensure that there's going to be a source of energy, there's going to be a carbohydrate source that the sperm can use to generate ATP. Another important component of seminal fluid is the presence of what we call prostaglandins. So prostaglandins are, we've talked a little bit about, but what prostaglandins do in the context of reproduction is they help to activate the sperm, and then they also help to so when sperm get into the female reproductive tract, there's a clotting process that occurs so that the sperm essentially forms kind of like a mucousy plug to help the sperm have a chance to essentially to swim up the female reproductive tract. And the prostaglandins also help to kind of soften and dilate the cervix, which makes it easier for the sperm to swim up into the tubes. And I guess the only other thing I would point out on this side is that this is where the seminal vesicles are located. So again, here's the vas or the ductus deferens, and then here's where you get the vast majority of the seminal fluid being secreted from. And then this next structure here is the prostate gland, and then here the bulbourethral gland. So this is just to help you kind of picture the relationship of these structures to one another. Okay. The prostate gland. So here's what I want you to know about the prostate gland. You only find a prostate gland, well, you only find all of the structures we've talked about so far, but just to point it out, because it's not always obvious to everybody, you only find prostate glands in males, right? Females don't have any of these structures. So what the prostate gland is it's this donut-shaped gland that circles around part of the urethra, right? And it's right underneath the bladder. The prostate gland, a lot of people are familiar with the prostate gland, especially genetic males, because if you go in, the prostate gland is something that is checked. The way to check the prostate gland is rectally, right? To make sure that it is, and the prostate gland is palpated to make sure that it's not enlarged. And that is because the prostate gland much to some degree like mammary tissue in females, but the prostate gland for males is very susceptible, especially as males get older, to developing their prostate cancer is relatively common cancer. And that's because in part, the cells of the prostate gland are relatively mitotic. So they undergo pretty rapid proliferation. And anytime you have a rapidly proliferating cell, you're going to have a structure that is more susceptible to potentially accumulating mutations and becoming cancerous. So prostate secretions are, I have here slightly acidic, but it's slight. I mean, it's still within the 7 range. But importantly, what the prostate secretions contain are substances that activate the sperm. So in the absence of a prostate gland, sperm are relatively sluggish, right? So the prostate gland kind of wakes the sperm up, helps to activate them. Seminal plasmin and PSA are 2 substances that help to do that. PSA, that acronym stands for prostate specific antigen. And this is the level, clinically speaking, PSA levels are used to help determine whether or not the health of a prostate. So when PSA levels go beyond a certain range, that can be indicative of prostate cancer. So it's a pretty useful marker that is relatively, it's a relatively non-invasive way to monitor somebody's prostate gland. And then the bulbourethral gland. So the bulbourethral glands, their secretions are relatively thick compared to the seminal fluid or the seminal vesicle fluid and the prostate fluid. They're kind of a thick mucousy type of substance. And from a functional standpoint, what these secretions do is
Uh, I think you're like cutting out really bad right now.
Can somebody give me back?
We can hear you now, I think.
Okay. Okay. So shoot, guys, when did it start getting sketchy for you all?
Just after you started talking about the bulbourethral glands.
Okay. So let me just say, so for whatever reason, this completely cuts out, I'll just go back in and log back in. Okay, so hold on. This happening again. Okay, so the bulbourethral gland, the secretions from the bulbourethral gland are thick, they're mucousy, and what these secretions do is they lubricate the urethra and just make it so that when semen is ejaculated, it doesn't kind of clot. These secretions also help to neutralize the pH of the urine so that the semen or the sperm aren't damaged when they're ejaculated. Okay, so that's kind of, I think I mentioned everything in here. Again, with prostaglandins, I do want you to know prostaglandins are really important, and we'll talk about prostaglandin, or you'll talk about prostaglandins a lot if you go into really any healthcare field, because they have so many different effects on a variety of different systems of the body. But in the context of talking about reproduction, they help to, like I said, kind of thin out, if you will, the cervix. So the cervix itself, which this we're now talking about the female reproductive tract, but the cervix has kind of a layer of mucus around it, and that helps to prevent pathogens and other substances from gaining entry ultimately to the uterus, because that's what the cervix is like, the neck to the uterus. So what prostaglandins do is, amongst other things, is they thin out that mucus. And so it makes it just easier for the sperm to gain entry, because they have to swim up through the vagina, which is a tube, through the uterus, and then into what we call the uterine tubes or the fallopian tubes. They also help to dilate the cervix a bit, which I don't have on this side, but they do. And then stimulate reverse peristalsis. So peristalsis, remember, is movement, is forward-like movement. And so, and we talked about it in the context of the GI tract, but there are other organs that have smooth muscle that undergo peristalsis. And so, and the uterus is one of them, right? In fact, contractions are kind of what the uterus does when childbirth takes place. So reverse peristalsis just means that the movement, instead of the movement going from anterior to posterior, or rather superior to inferior, so rather going in a movement that's kind of top to bottom, the uterus does this kind of reverse contraction. And what that does, functionally speaking, is it just helps to kind of create a vacuum to pull the sperm into the uterine tubes. Okay, and then the other enzyme I have mentioned here is fibrinolysin. So fibrinolysin is an enzyme that helps to essentially, as I mentioned earlier, semen itself kind of forms a clot. It has coagulation properties, somewhat similar, I suppose, to blood, or you can think of it that way. And that helps so that the sperm kind of are able to sit in the female reproductive tract and start to swim, up toward the uterus. And so at some point in time, obviously this clot needs to be dissolved, and fibrinolysin is an enzyme that helps to do that. And then lastly, I guess I mentioned this already, but if sperm are not ejaculated, they are reabsorbed every 2 weeks by the male body. Okay. And then this is just a slide that you can use to help you study. Here's some, different histological examples of what these structures look like under the microscope, but I'm sure you're kind of covering that in pretty good detail right now in lab. And then finally, when it comes to secondary sexual characteristics, here's what I want you to know. So post-pubescently, during pubescence, that's when we see that's when secondary sexual characteristics occur. And so what these include for both males and females is the growth of hair in the pubic region, in the axillary region, and then for males in particular, on the face, right? And that's because of the influence of testosterone. Testosterone, there are lots of cells in the body have receptors for testosterone. And we've talked about many of them throughout the course through 227 and 228. So bone has a lot of receptors for testosterone, as does muscle, there's lots of receptors for testosterone on the laryngeal plate, right? Which is why as males go through puberty, voices crack to a greater degree than what you see when females go through puberty. And that's because there's this huge, there's a growth or a widening and thickening of the vocal cords, and then the development of the Adam's apple, which is the laryngeal prominence. All of that is because of testosterone. So really, testosterone is responsible for all of these secondary characteristics, sexual characteristics that we see in males. And then, as I mentioned, it's the libido or the sex drive hormone for both males and females. And then you can use these slides. I'm not going to ask you any anatomical characteristics or features of the penis. Most of I left this on there because you can use it. I think it's, anyway, because I used to discuss it when I would do this with lab, but I'm going to leave this up to lab now. So I'm not going to test you on that side. Nor will I test you on this one, but I do think this is a good image and you do need to know it for lab. If you were to do a cross section through the penis, right, this is what it would look like. So we have what we call the corpus spongiosum, and then the paired corpora cavernosa, right? So what you can see here, and it's through the spongy urethra. I'm sorry, it's through the corpus spongiosum that you have the spongy urethra that runs, right? And this is so the urethra, basically. Okay, and then finishing up with male reproduction. So when it comes to the sexual response, and this is what I want you to take away from this, I want you to think of it in terms of the role that the autonomic nervous system plays in reproduction, right? So when we talk about the male and the female sexual responses are similar, but there are some, some important differences. So for the male sexual response, the first phase is what we call the excitement phase. And the excitement phase is under the influence, or I should say that it's the parasympathetic branch of the autonomic nervous system that is responsible for the excitement phase, right? What the parasympathetic nervous system does in the context of the male sexual response, excuse me, is it causes vasodilation to the penis. So that's what's responsible for erection. It also increases the release of nitric oxide in that general within the cells, within the cells that make up the tissue of the penis. And again, nitric oxide is also a very potent vasodilator, right? So the take home is parasympathetic nervous system equals increased blood flow, okay? And then ejaculation is a part of the phase that's called, the phase itself is called orgasm, right? So, and the branch of the autonomic nervous system responsible for orgasm is the sympathetic branch, okay? And something that you should kind of be taking away right now is that a disruption in either sympathetic or parasympathetic nervous system control is going to make reproduction difficult, right? It's either going to compromise the ability for erection to take place, or if the sympathetic nervous system branch is damaged, it's going to compromise the ability for ejaculation to occur. And then the last response is what's called resolution, which is still under the influence of the sympathetic nervous system. And during resolution, or I should say shortly following resolution is just the reestablishment of pre-excitement physiology, right? And then right after resolution, there's what's called the refractory period. And the refractory period is this period of time during which the parasympathetic nervous system cannot be stimulated to an extent that would cause excitement or erection, right? Refractory periods as men age get longer and longer, right? And then an important difference between males and females is that females don't technically have a refractory period. Yeah. And then climacteric. So male climacteric is kind of the functional equivalent of what is called menopause in women. So menopause in women is this event that after which females are not reproductively capable because they no longer have eggs to ovulate, or they're no longer ovulating. Whether or not there are eggs there that are viable is kind of still a field of research, but after menopause, females are no longer ovulating. The equivalent of that would be this, what we call the male climacteric event, but it's not really an event. It's just this process that stretches on throughout the duration of the male life, the lifespan. And so just like females, what happens is after, and it's the ages line up pretty closely. So at about 55, which I think is the average age for menopause to be complete in women, at about 55 for males, you see climacteric types of issues. So fertility for males takes a nosedive. Males at that age start to encounter, erectile dysfunction disorders much, much, much more frequently. Um, you know, all that fun stuff. Okay, so let's see what time it is. So I guess we'll stop. Like I said, we're not in a huge rush. We can do Tuesday female, and Thursday we'll finish up with the development and heredity material, which I think that I have, I don't know, maybe 35 slides to get through with that.